Hello students, today I am going to start a question paper discussion subject Biology class 12 Revisionary examination 2025 to 26 Sibsagar district. First part Botany. First question, what is geitonogamy? Geitonogamy is a form of pollination where pollen is transferred from anther of one flower to the stigma of another flower on the same plant. Next question, why selectable marker is an important feature of a vector? Selectable marker is an important feature of a vector because it allows scientists to identify and isolate the host cell that successfully took up rDNA transformation. Next question, if the chromosome number of the same somatic cell of an angiospermic plant is 24, what will be the chromosome number of endosperm cell of plant? First, the endospermic cell is a formula trishan, that is the n plus twishan, where twishan is equal to 24 in angiospermic plant, then n is equal to 12, then trishan is equal to 36, that means 3 into 12. Next question, give an example that indicates homology. Homology actually indicates that the same origin but it creates different functions. For example, thorn of bougainvillea and tendrils of cucurbita. Next question, name two antibiotics produced from fungi. Two antibiotics produced from fungi are penicillin and cephalosporin. Next question, which type of ecological pyramid is allows, upright and why? Ecological pyramid is always upright in case of pyramid of energy actually, and is because energy flow is unidirectional in the food chain, which is decreasing at each trophic level by 10% law. Next question, explain briefly detritus food chain. So, detritus food chain, it starts from decayed or dead organisms from the decomposers which break it down by some microorganisms, bacteria, fungi, etc. Next, trophic level. Actually, trophic level is the organism's position in food chain, where for example, producer, which is trophic level is L1, then first primary consumer trophic level L2, then secondary consumer trophic level L3, then tertiary consumer or topmost consumer, that is L4. Next, secondary productivity, it is actually a rate which consumes create new organic matter by eating other organisms. Next, write the scientific name with their utility in the following plants. First, neem. The scientific name is the Azadirachta indica, then family is Meliaceae. It is a medicinal plant. Next, teak, scientific name Tectona grandis, family Lamiaceae, it is actually timber yielding plants. Next, sisui is also a timber yielding plant, and the scientific name is Dalbergia sisu and family is Fabiaceae. Then tulsi is a medicinal plant, scientific name is Ocimum sensititum, family is Lamiaceae. Next question, what is sewage? Okay, and in which way it is harmful to us? Sewage, it is actually wastewater from homes, industries, drains, then streets, which contain some organic matters like nutrients, chemicals, okay, then also some harmful disease causing microorganisms like pathogens. And these pollutants waters spread some disease like cholera, typhoids, okay, it also creates some algal bloom. Next question, explain briefly the role of microorganisms in sewage treatment. So, sewage treatment microorganisms, some which help this treatment. Next question, how is genetically engineered insulin produced? So, in genetically engineered insulin produced by inserting some human insulin gene into a bacteria or some fungi also using rDNA technology. Next question, what is genetically modified crops or GMO? GMO with a contrasting of two plants, one is the host plant, then another one is hybrid, okay, which produce some GMO, for example, broccoli, okay. How are GM crops different from hybrid crops? GM crop is very disease resistant, some pest resistance, and it is easily available in markets. So, these are very improved than this hybrid crop. Next question, what are molecular seizure? So, molecular seizure, it is actually enzymes that out DNA as specific sequence, okay, it cut out, for example, E. coli, BAMH1, okay. Then next question, what is bioreactor? So, bioreactor, it is actually control and closed vessel where some living cells and also enzyme transform into medicines like some beneficial products or fuels also. Okay, then next question, describe briefly the process of development of female gametophyte in angiosperm. So this female gametophyte develops from this some single functional megaspore, which the ovules is by 3 mitotic divisions, which is produced to form 8 nucleus or 7 cell structure, and the egg cell, 2 synergy cell and 3 antipodal cells, and also a central cell, which is produced from by fertilization. Okay. Next question, discuss the development of dicot embryo. So dicot embryo is the starch which is zygote, which is dividing into 2 cell pro-embryo containing basal and apical cell. Next question, what is state the Hardy-Weinberg principle, list the factors that influence a law. So Hardy-Weinberg principle is a allele and large genotypic frequency randomly mating population remain constant, where the formula is p square plus twice pq plus q square is equal to 1. Where no disturbance is produced, then the no variation is formed. List the factors that influence this law. The factors are like mutations, like natural selection, then genetic drifts, and gene flow, etc. Next question, explain adaptive radiation. Give 2 example of adaptive radiation. So adaptive radiation, it is actually evolutionary process where single ancestral rapidly forms or diversify into many new species. For example, Darwin's finches, then Australian marsupials. Okay. Next question, explain the steps of recombinant DNA technologies. So the rDNA technologies is formed by isolation of genetic materials, then cutting DNA, then isolation of desired fragments DNA, then ligation, then insertion into host, okay, then selection and screening test, then the cultural and expression, then downstream processes. Next questions. This is the last question of botany, that is, they explain the process of agarose gel electrophoresis, state the application of gel electrophoresis. So agarose gel electrophoresis is a separation technique. Okay, separation of DNA fragments by size by using electric field where positive and negative charge is present. Then its the state's application is the genetic fragmentation fingerprinting uses, then disease diagnostic uses, then gene cloning also used. Next part B, zoology. First question, ovulation is induced by hormone, LH or luteinizing hormone. Then second, the disease transmitted from sexual intercourse is called, that is the sexual STDs or STIs, sexually transmitted infections or sexually transmitted disease. Then AUG codes for, it is the amino acids methionine or start codon. Then typhoid disease is diagnosed by Weidel test. Next question, what is ovulation? So ovulation is a ovary which is released from mature egg or ovum into this fallopian tube. Next, what is template strand? Template strand, it is a antisense strands, is one of the 2 DNA strands that serves as a guide for DNA polymerase to build a complementary mRNA during transcription. Next question, what is transgenic organism? So transgenic organism is a living thing whose genetic material has broken altered by the introducing a foreign transgene from different species. Next, name one World Heritage Site of Assam. So World Heritage Site of Assam actually is Kaziranga National Park, then Manas National Park, and also is Raido Moidam. Then question, what is copper-T? Write its uses. So copper-T, it is actually a STD or IUD, intrauterine device, which is a hormonal free birth control contraception. It affects actually motility and also sperm lysis. Okay, and from this the fertilization is not occur. Then write the application of DNA fingerprinting. To DNA fingerprinting, it is actually applied in the forensic science or criminals test or paternity or maternity test, also identification of missing person, then medical diagnostic uses, then also wildlife conservation and evolutionary history. Next question, what are the barrier of innate immunity? So innate immunity, it is actually is a barrier which is like physical, like skin and mucous cilia, and physiological methods like the tear or saliva or some hydrochloric acid which is produced in stomach, then also sweat glands or sebaceous glands, then cellular some methods like neutrophils, macrophages are produced, then cytokines where interferons are produced. Then question, define ecological pyramid, give examples. So ecological pyramid, it is actually graphical representation or model which showing in biomass or number or energy at successive trophic level in an ecosystem. For example, grassland pyramid of numbers. Then question, write the scientific name of Muga silkworm. Actually, it is a Anthurium samensis, and the mention to host plant of the Muga silkworm is a Persia bombycina, which is sum, and the Lichia monopetali, that is swallow plant. Then distinguish between incomplete dominance and codominance. Actually, incomplete dominance, it is a create in absent. Okay, and it is, for example, red color flower and white color flower to produce a pink color, that is the indeterminate form, that is it is the middle form of this male flower and female flower, that is pink flower is produced. It is then the codominance, it is show both traits, that means it is distinctly or separately both male and female character is produced in the F1 generation. For example, ABO blood group. Okay, then question B, transcription and translation. So transcription is a copy of DNA which is produced to mRNA and occur it is in eukaryotic nucleus. Okay, then translation, it is actually decode of mRNA which produce protein at ribosome in cytoplasm. Then question number C, vaccination and immunization. So vaccination is a actually action which is getting a vaccine. Then immunization is a result or which is body build protection, and this build protection is from antibody, and this antibody is produced from this vaccination, and also it is produced from naturally by inborn. Then draw a level diagram of TS of ovary. Then draw a level diagram of Watson and Crick model of semi-conservative replication of DNA. Then question number 6, write briefly about the disease of silkworm. So disease of silkworm like viral disease Gressari, then bacteria or viral disease like Flessari, then fungal disease like muscardine, then protozoan disease like pebrine. Okay, then write various reasons of loss of biodiversity. So loss of biodiversity causes like habitat destruction, deforestation, urbanization, overexploitation, introduction of invasive species, then climatic changes. Next question, write the salient features of human genome. So human genome, it is actually 3 billion pairs are present in this gene, and approximately 20,000 to 25,000 genes with large repetitive sequence are present, where 99.9% are identical and only 0.1% is the different, which is actually is called DNA fingerprinting. And the people which know in chromosome 1 has the maximum gene, and the chromosomes Y which is present in male gene is least gene. Okay, then question number 8, describe the various chromosomal disorders. So chromosomal disorders, it is Down syndrome, Turner syndrome, okay, Klinefelter syndrome, then deletion, addition, monosomy, trisomy, translocation. These are all about the disorders of chromosomal. Then describe about the pedigree analysis. So pedigree analysis is a genetic tool that studies the inheritance of the trait through a family history by sort using symbols, some maps, and generation. This is actually help in determine to a trait it is dominant, also recessive, autosomal, and sex-linked disorders. Next question, describe the structure and function of ecosystem. So ecosystem, it is actually involved in this biotic component like producer, consumer, and decomposer. Also some abiotic components like sunlight, water, temperature, pH, soil. Okay, and these are actually the interaction in between the biotic and abiotic species, okay, which produce a forming interconnection wave by energy flow and nutrient cycling. Then last question, describe the mechanism of transcription. So transcription, it is actually the formation of the DNA, copying DNA, genetic codes into the mRNA by interaction of RNA polymerase binds to this promoter, then which is called initiation process, then the elongation process where the complementary RNA is formed, then the termination process where hnRNA or heteronuclear RNA is formed, where both exon and introns are present, and after splicing the only nuclear RNA is produced in the nucleus, which later transcribed into the cytoplasm after translation. Okay, so these are all about the discussion of the paper Revisionary Examination 2025 to 26 paper Biology class 12. Okay, so thank you.